drug target and repurposing prioritization. And so for that, we have a couple of funded projects in cancer and rare disease for nominating drugs to treat patient cellular phenotypes with different computational approaches and test-generated hypotheses with collaborators. And so we have some, as I said, some projects in the cancer space, as well as through the Center for Precision Animal Modeling that I'm a part of here at UAB. And finally, really about cell and tissue specific context dependency. So trying to investigate the impact of gene regulation and expression, genomic instability, cell-cell signaling, and age-associated molecular changes on disease manifestation. And so we kind of view ourselves as really being both well-versed in wet lab and dry lab genomics and data science techniques, and really trying to sit on the bleeding edge of what's happening in those spaces. And one of the things that became really apparent from being here at UAB was there's kind of a hunger and a need to extend some of these different approaches in other labs in a way that is, you know, outside of what we can do ourselves. And so as part of that, you know, myself and some of my colleagues here were really trying to think about how we can make some of these different approaches and techniques more accessible to the community. And it's really kind of broadly under this big broad umbrella of genomics. So here is a figure I made that I like to show, really thinking about genomics as encompassing kind of all the things within this, you know, across the central dogma of biology. So everything from variants and copy number aberrations to methylation, DNA protein interactions, protein abundance, RNA messages, methylation, and even beyond that, really trying to understand chromatin conformation and other aspects of genomics. And so, you know, our goal here at UAB broadly is really to try and identify this genomic variation that's associated with cancer or disease to improve patient care. And broadly speaking, you know, there's a lot of groups here that really want to incorporate genomics and data science techniques into their research. And so that has become even more possible here in the last couple of years with the drop in sequencing costs. So everybody's sequencing now and really trying to figure out what do we do with that data and how do we analyze and interpret it. And so I, you know, just wanted to show as an example, you know, the amount of data that's being generated in the community broadly. So not even here at UAB, but broadly. So if we take the Cancer Genome Atlas as one example, you know, this is hundreds of thousands of data files, over 2.5 petabytes of data. And it's an insane amount of data. So we have, you know, if we take that 2.5 petabytes of data and translate it into something that's maybe a little bit more accessible for everybody, you know, this is an insane amount of data. So about 50 million 4-drawer filing cabinets have printed, 5,000 years of MP3 songs, estimated as the human brain's memory storage capacity. So it's really a lot of data that has begun to permeate modern biology. And so that's really where my group and my colleagues who are co-founders of this core kind of come in, is that's kind of our expertise, is this intersection between biology, computer science, and mathematics. And so it's kind of coined this idea in the field broadly that we're not really talking about astronomical data anymore. Kind of the dad joke here is that we're talking about genomical data and how do we analyze that and how do we do that in a way that's accessible to more research labs here at UAB. So here's a figure from a paper that I pulled that's a few years old now. And the idea here is, though, trying to project out over the next decade, what amount of annual data storage do we think we'll get? And, you know, the punchline here is genomics is going to outdwarf Twitter and YouTube and astronomy, hence the genomical, not astronomical data. So, you know, again, this has become pervasive in kind of the field of biology, and modern biology is this big data generation and really trying to understand what's happening in a system on, you know, a cell by cell basis, and be able to interpret that in order to learn about the biology and help patients. And just to really hammer home again, the need to have experts in the area as part of, and in the room during planning of these sorts of experiments. Here's a paper that just got published in the last couple of weeks, remarking that there's over 1,000 tools that reveal trends in single cell RNA-seq data. And so trying to understand, which of those tools are the most appropriate for a particular study design, and trying to understand how to implement them to answer a research question is just really, really challenging if your expertise happens to not be in computational biology. And so that's really where we come in. And that's the premise behind the core. And our director is Dr. Liz Worthy. She's an associate professor over in Peds and Pathology. Myself, and then Dr. Lara Ionov, who is a senior bioinformatician in the Civitan, and she's the managing director. And so our goal here is to provide this research resource, providing services to the community to identify, characterize, and quantify molecular variation and impact, to provide exceptional computational analysis and method development support, computational services with an emphasis on genomics, transcriptomics, systems biology, and translational medicine, and provide training for biomedical investigators. So the three of us kind of sit with our groups kind of at the crossroads of these different areas, and a way to scale that and help move more research forward here at UAB. So I want to give you an example of some of the current services that we're offering, and kind of show you, what that looks like, and talk about, if you come to the core for as a user, what that process is like. So kind of our most popular service by far is transcriptomics. And under that umbrella is RNA-seq, microRNA-seq, single cell or single nuclei RNA-seq, site-seq. All of these fall under that umbrella of transcriptomics. And so one of the things that we've done is really try to standardize the reproducible research aspect of transcriptomics. So here is an example of a report by Dr. Ionov using our bulk RNA sequencing pipeline and analysis. And I just want to note, one of the things that, we're all scientists ourselves. We all write grants, we all publish papers. And so we're very mindful of doing reproducible research and making sure that we stand behind and feel good about the data analysis that we're working with you on. So here's an example of a report. This is an example from the Day Lab. So for part of this, it includes a PDF and an HTML report, including as well as all the different file outputs about differential expression and figures and raw outputs as well. But we also have this report, which I'm showing you now, that comes with it. And so, for example, we document all of the upstream data analysis and have all of the versions, and really explaining through what the different parameters were that were used for things like data alignment, and explaining everything that you would need in order to be able to write up that method section and to feel good about the analysis that's going into your research project. Again, all of the details of what the pipeline configuration is, so that you can go back and see exactly what it was that was used to analyze your particular data set. We run standard quality control tools. So for example, multi-QC. And if this were, I took a screenshot, but if this were the HTML, you'd be able to scroll through and you'd be able to see all of the different quality control metrics for individual samples that are part of the project. And also statistical results and downstream quality control. So for example, here's kind of the exploratory data analysis stage, where you can see what the principal components analysis looks like. And we've colored them here based on particular experimental attributes as well. And again, just additional examples of some of the exploratory data analyses that come standard as part of the RNA-seq workflow through the core. So this is an example of sample to sample distances. One of the, probably the most popular analytical requests for transcriptomics is looking at differential expression analysis. So in addition to the full outputs from vetted tools like DESeq2, there are publication quality graphics like this MA plot that shows differential expression. And again, includes all of the details of how the analysis was done, so that you can report those as part of your project, and all of the associated files that are, clearly named and have all of the parameters included there as well. We also will include common figures. This is something that during our consultation, which is usually Dr. Ianov, will sit down and talk with you about what your specific needs are for your project. So common figures include things like volcano plots, heat maps, or other gene level expression figures. Additional analyses, things like Go enrichment analysis are also included as part of kind of the standard RNA-seq pipeline. And then we can also work with you to add additional requested modules for transcriptomics. So again, just as an example of kind of the sorts of analyses that you can, come and get help with through the core, and the kinds of outputs that you can expect to receive back. And so that includes all of the raw data, the analyzed data, as well as a final report that includes all of the parameters and walks through the analysis and those analysis steps. One of the other kind of popular services for the core is interactive web apps. So this is something that a lot of folks really like, because it allows you to be able to take a look at the data and ask questions in the data without having a programming background. So this is something that we highly recommend to customers who come, or users that come through the core. So here's an example that's part of the same project that I just showed you from the day lab. They had a rat atlas, brain atlas, so they called it the Ratless, that Dr. Ianov built for them, and you can go play around with it right now if you like. If you go to day-lab.shinyapps.io/Ratless, you can go play around with it yourself. Dr. Ianov included information about how the data was generated right there on the app to make it easy for users at other institutions or in other labs to be able to kind of see what exactly it is that's been done, as well as all of these are interactive. If you go to the Shiny app, where you can ask questions about coloring genes from different, this is a UMAP plot for single cell data. So ask questions about how much a gene is expressed in particular cell types. There's a whole bunch of different customization options that are part of this interactive web app that users can use. So this is really nice if it's a data set that your lab has generated and you're using to ask questions a lot, and you want to be able to come and be able to work with that data and look at it. Thank you, Sharita. Sharita put it in the chat for you guys if you want to go dig around. So, thanks to Jeremy's lab, they're real good sports about letting us use their work as our public facing, so people can see what it is that we can do. But you can click around and look at different experimental conditions, as well as color different genes and make violin plots, all sorts of different plots here. So again, this is a really nice option to add along to an RNA-seq analysis as part of a publication, so that other people can come and ask questions in your data easily, or just to have for your lab to be able to access the data more easily. So an additional service that we offer in the core is genome sequence analysis. And so I want to show you a couple of examples of the sorts of analyses and visualizations that can come with that. Similar to transcriptomics, there's data QC analysis and prediction. So for example, we do offer services to look at prioritization of variants from patient genomes. So this is part of the Codicum software that was developed by Dr. Worthy's lab. We also offer where we can do multi-sequence alignments and also look at different protein folding and ask questions about variants in proteins, variant interpretation and classification. So that's something that we also offer as a service, is looking at scoring variants from patient genomes and asking about ACMG classifications like the pathogenicity of those variants, as well as data integration, comparison, and visualization. That's something that the core has a lot of experience in, as custom visualizations and custom data integration. So that's something that we can meet with users on a case-by-case basis and work with them. I wanted to mention a couple of other, like, one of the things about data science and genomics in general is the field just changes. The field just changes so rapidly that just because we don't have services listed on our website doesn't mean that we're not able to perform those services. So we have a whole slew of custom services that we're working on right now. And so I just wanted to give you a couple of examples. So one is a project-specific Docker container. So Docker is basically allows you a box to put all of your analysis in and your tools in and your compute environment in, so that other people can easily reproduce what you've done. And so some funders are now starting to move towards requiring those sorts of elements for work that is produced from their funding. And so that's something that we offer as a service to help you with, is to wrap those up. It's also really nice to be able to publish your paper and include that Docker container so that other people can go and easily reuse or reproduce your analyses. Another area that we currently have as a custom service is we're working on implementing a nanopore pipeline. So this is for long read sequencing. And so we're developing that pipeline here at UAB for use with research projects. Another example is data integration. So we have several projects that are actively integrating 10x multiome analyses. So this is single cell data from using both RNA-seq and ATAC-seq data. And so those are projects that are ongoing in the core and are something that, as these new integration techniques, or as these new wet lab techniques come out where we're able to assay multiple data types from the same cell, that's something that we are offering as a service in the core, is to help with that data integration. And then wrangling public data sets for comparison to locally generated data. We have a couple of active projects in the lab now where we're using different neurological or cancer data sets and helping research groups wrangle that data so that they can use those as orthogonal validation or to generate hypotheses for testing in their own lab. And so that's something as well that we are able to offer as a service. And then finally is really study design and figure development for proposals and manuscripts. So one of the things that I think is key for really thinking about using genomics and data science as a core service is to include the cores early and often in your proposal and manuscript preparation, because we can help you with things like power analysis. We can help you with making sure you have the latest and greatest tools. We can help you with trying to make sure that you scope out your project appropriately for the analytical costs. And so that's something that we also offer as a service, is even if you don't have your data yet, is really trying to understand like how you scope out your project for that next proposal, and et cetera. So we, again, genomics and data science is a quickly evolving field. So it's something that you can contact us and we can discuss if it's a good fit for the core to be able to take on that particular custom work and help you move your science forward. So that hopefully gave you kind of a feel for some of the different sorts of services that are offered through the core. I want to talk about kind of the other main arm, which is training and outreach. And so we have several different training and outreach programs under the UBDS umbrella. So the first one is I and folks in my lab have developed data science guides, and we're going to continue to develop these as part of the core moving forward. So if you go to my lab website here, you can take a look at our education panel, and we have several different data science guides, and I'll show you an example of those here. So for example, this is our R master guide. And so we view this as vetted resources. So if you've never programmed with R before, but you want to learn how to program in R, this is how we recommend that you get started. And so we have some information for how to download R and R Studio and vetted instructions for how to do that. And then we have a couple of different online free resources that this is how I train students who come through my lab to learn how to program in R. So Swirl is an interactive R programming that teaches how to start kind of from the very beginning with no programming experience and be able to kind of move through things like basic building blocks all the way through matrices and data frames, functions, apply functions, and even into working with some base graphics. And then as well as the software carpentry. So I am a certified software carpentry instructor. This is a group that is an international group. It's a nonprofit, and what we do is we teach folks how to teach data science and programming skills for non-data scientists and non-programmers. And so it's a very thoughtful set of coursework for being able to do that. And so there are links here to two of my favorites, R programming courses that, again, all of this is free, but as part of that R master guide, so that, if a new grad student who's listening, or, if there's a faculty who has a new grad student in the lab and, this is something that, that student needs to learn or you need to learn as part of your project, this gives you kind of a place to get your feet wet using resources that are vetted, and that there are other people here at UAB who have experience using those training methods. And so to complement that, it's great to, sit with a computer by yourself and be learning how to program or learning about data science, but to complement that, I have a UAB Labs Slack community that you can join. I can make these slides available, or we can, I can try and pop this in the link when I'm done with the talk if I remember. And so this is a community that, again, is free. It's a messaging app, and we have a data science channel there, and myself and others from the core monitor it. And we try really hard to make sure that everybody gets some sort of response within, a day or two, helping you find a next action. So that could be, as simple as I'm going through this, R tutorial and I've got this error, I'm stuck, to something more complicated about a particular data analysis. And it has, I just checked yesterday, and there were I think there were 116 folks in the channel. So it's a pretty robust group of burgeoning data scientists here at UAB who are part of that community, and really trying to give everybody a place to say, hey, has anybody used this tool before? Does anybody have any insight on this analysis? I have this weird graph. I can't get this error worked out. I saw this cool paper. I thought you guys might think it's cool too. There's kind of a lot of different discussions that take place on that channel, but it is a place where you can reach out to other people here in the community that are thinking about the same sorts of problems, and form some relationships. So everybody who has a UAB, or I think I've tried to hit all the like .uab, UABMC, all of the email addresses, you can join yourself with the link. And so we'll make sure that that's available to you later if you'd like to. again, kind of moving from the most independent to the most hands-on, the next step is data science office hours. So myself and Dr. Ianov run a data science office hours every Thursday from 3:30 to 4. There is a registration link that you sign up for in order to join. It's also pinned in the data science channel on Slack, or you can email me and I'll help you get the link. But we sit there every week and we do our best to help you with whatever your next action is. So both Lara and I have extensive wet and dry lab backgrounds. So if we know anything at all about what you're doing, we will help you figure out a next action. If we're not the right people, our goal is to help you find the right people here at UAB so that you have that as your next action. So everything from study design to comparison of tools and algorithms, to kind of really anything. It runs the gamut. We've had I think almost 100 folks come to office hours over the past year and a half. So it's a great place to come and also talk through. We've had students come and say, hey, I have this really bit for my qual that I'm not sure about. Can I come and talk to you about it? And so they'll come and sit in office hours. On weeks that nobody comes with questions, we talk about cool papers or, Twitter flame wars on computational biology. Really try to use the time to at least talk about data science. We have several people who come and just sit and listen. Even if they don't have specific questions that week, you kind of learn something by being in the room and hearing other people ask questions, and that's totally fine too. So we're there every 3:30 to 4 on Thursdays. If we do cancel occasionally, like next week for Thanksgiving, we do post that on the data science Slack channel. That's how we communicate with the group. So again, that's kind of a hands-on weekly where we're there. It's free. You can come ask questions. And our goal is that you leave with the next action to keep you busy before the next week's office hours. And again, moving kind of from self-directed, the guides, to the Slack community, to office hours, kind of the next rung in the ladder for training and outreach is workshops. So we are offering workshops. Again, we're a new course, so we've offered one workshop thus far, but we'll be offering more in 2022. And so I thought I would kind of walk you through what our first workshop looked like, so you kind of know what to expect when you see new workshops pop up in the future. So our first workshop was held in August, and it was titled Programming with R, and it was a 2-day hands-on intensive training event for UAB students, postdoc staff, and faculty. I will say I've gotten asked this question several times. While it was majority students, there were several postdoc staff and faculty there at each of those levels. So it wasn't only students. And myself and Dr. Ianov, and then Dr. Oza, who's a senior scientist in my group, we took turns teaching from the front of the room, but it was all hands-on work. So we would live code and folks would code along with us, and it was meant for people who had no programming experience. And then we had a group of intrepid students, including some of the ones who are here, who are part of the Informatics Club, who walked around as helpers at the workshop, and we kind of had a leave no person behind policy. So, if you're stuck or it doesn't make sense, it's no big deal. We had helpers who would come around and one-on-one work with you and make sure that, it was a good use of your time and that you could get out of it what you needed. So we covered all of these different topics about introductory programming. It was pretty successful. So it was free. It was our first one, so it was free, but it sold out in the first couple hours, and we had a really long wait list. The feedback that we got on the workshop was very, very positive, and this is something that we will be offering more of here in 2022. But the material was based on that material that came from those data science guides that I showed you a few slides ago. So it is something that you could go and work through yourself, and then if you had questions, you could come to the data science channel or to office hours and you could get help. You don't have to wait for the workshop, but we will be offering more workshops in 2022. So before we, wrap up today and then make sure there's time for people if they have questions, I wanted to point out a couple of resources that are not part of UBDS, but are just part of this data science community and this genomics community here at UAB. So I mentioned kind of here at the top, this genomics and data science. This here is us. So the institutional research core, which you can go check out our website, the guides, which are on my lab website, the Slack channel, office hours, and then the workshops. So those are the things that we just talked about in the last section. I do want to point out that our good friends over in research computing also have office hours. So if you are having trouble with navigating Chiaha or those sorts of questions, moving big data around, they also will do their best. Just like we do our best, if we know something about it, we'll try and help you. They kind of have the very similar mentality, so they'll try and help you with programming questions. And they have office hours typically on Mondays and Thursdays from 10 to 12, and I believe they're on Zoom right now, though they're talking about moving them back to in-person or doing a split thing. They also have monthly training sessions at the link above. They're usually somewhere between, I think they're an hour or two or an afternoon once a month, and they'll do things like how to use notebooks for research computing, or how to move data using Globus, or how to access Chiaha and move around. And they're pretty good about letting you know what it is that you need to come in and get the most out of it. And so keep your eyes out on that. You can get on their mailing list as well. And then there is also a journal club, data science club, that is great for the students. And they do a lot of like pseudocoding and kind of talking through these different problems. So that's something that might be of interest to the students in the room. If you have a more biostatistics question, like a clinical trial, I want to get a p-value, those sorts of questions, there's the Biostatistics, Epidemiology, and Research Design CCTS program that you can reach out to, and they would be happy to sit down with you and kind of talk through more biostatistics and clinical research study design, and you can check that out there. The other thing that I thought I would just mention is that I teach 2 classes. So if you're looking for hours and you're a student, in the fall, I have a journal club called Genomic Science Applications and Disease Research. Several of the students on the call here today are in there, so you can go talk to Sharita or Tarun or Ari. They're in the class right now if you want to hear a little bit about what that's like, or go talk to the students in my lab. And then in the spring, I will be teaching the Genomic Sciences course. This is the GGB theme core class, and it's in February roughly. And so I will be teaching that starting here in 2022. So how can you get a hold of us? So you can email Dr. Ayanov directly. Here is her email address. We also have an inquiry form on the website. And we have our own Twitter handle as well that you can check out. So the way it works when you reach out to Dr. Ayanov or on the inquiry form is you'll be directed to fill out the inquiry form. We receive that information. We'll reach back out to you with any additional information we need. And then it's just part of the initial consultation to get to know each other and kind of figure out what the next steps are. We will schedule a meeting with you to go through and talk about like, what is it that you actually need and want, answer questions that we have, we answer questions that you have, and then Dr. Ayanov works up what we call a scope of work. And so that is a document that really details like, this is what you're looking for, this is how we can help you, and this is, what the prices are for that. So office hours are free, data science channel is free, data guides are free, the initial consult is part of just being here at the UAB community. And then that scope of work will detail then how much, what the cost is for our personnel time to be able to execute those analytical steps. And again, we see it as a very iterative process, so we can continue to work with you and collaborate with you on that. And we do have both fee for service as well as like percent effort on grants as part of that. And for certain programs, there's also some additional pots of funds that may or may not be available depending on what program you're part of for pilot data and things like that. So I think I will end here so that you guys, thank you, Tarun. Tarun posted the Slack space for you guys. And so I will end that here. I see Tarun had a question about for the core to offer courses for credit. As far as I know, there is some talk about having a course through that would be kind of with a cancer perspective that would offer an opportunity for folks to be able to kind of walk through some of the different cores and learn about different techniques in the cores. As far as like offering classes for credit through the core, not that I'm aware of. For me, I'm a professor, assistant professor anyway, so I teach classes outside of my role as the core, but certainly they all kind of feed back into one another and part of the genomics and data science community here. If there are other questions, I'm happy to, let me put the, that back up. I'm happy to talk to you about them. And hopefully this gave you an overview of what the core is, what it does, and how you might be able to work with the core moving forward.
Thank you, Brittany. Are there any questions? You guys can unmute yourselves or you can post it in the chat.
So Brittany, I thought that was really cool to see all the infrastructure your team has built. Can I ask perhaps a slightly obnoxious question?
Sure.
For those of us who hang out in the clinical departments,
yeah,
and are dealing with often much smaller data sets, is there any opportunities for help or collaborations for our folks?
Absolutely. So we actually have several users who are in the clinical departments and do have, smaller and bigger, I guess, is the eye of the beholder, and really depends on your frame of reference, but I know what you're talking about. And so absolutely, it's really more about the techniques and less about, I guess, the size of the data. I think the size of the data is usually, in my experience, what prompts the basic researchers to come seek help. As the data has just become so big, it becomes unwieldy and requires that extra help. But for the clinical researchers, absolutely. So there's no data set too big or too small. One of our charges, I guess, is in our, professional opinion, there can be limitations with smaller data if you're trying to analyze that by itself with respect to power or, limitations on what kinds of statistics can be done, as you know. And so that's something that actually goes into that scope of work document. And so we talk about that before any, money is charged or anything like that, is like, from our calculations, we think these are going to be limitations in the interpretation. And so we lay all that out in the scope of work document so nobody's surprised on the other side. So, yeah.
Thanks.
Yeah. Any other questions? I think it's like pretty straightforward. If you have, genomics or data science questions, there's lots of ways you can reach out to us by email, through the data science channel. You can drop by office hours. You can send an inquiry form. And again, if we're not the right people, then we will do our best to try and help you find the right people. But our office hours is really useful for the students. So I would encourage the students, if you're stuck or if you're just feeling a little alone, I know there's a lot of labs where you might be the only who's working on, that kind of data, then come check it out. So Tarun's volunteering to pay for everybody's services. Is that what you mean, Tarun? So the cost, we have a cost sheet and it depends on the scope of work. And so that's part of that contract, is working out, you know, how many hours it is for a particular analysis.
I had a quick question for you, Brittany. In terms of sort of workshops or sort of training, you know, what do you guys sort of have in mind? Obviously, R is pretty popular.
Yeah.
Do you guys plan to do anything that's more like, I guess, specific, like maybe an analysis type workshop or like?
Yeah, so that's definitely something that we are talking about. So, you know, one of the things, you know, as far as like other programming languages, I know that Research Computing recently had kind of a crash course Python workshop. And so that's something that we're cognizant of and like trying to like coordinate with them. That was kind of like, we did R, so they were like, well, we'll do Python because there was a call for Python as well. And so that's something that we'd like to continue to coordinate with them on in the future. So Blake, for those of you that know, Blake was really nice and came out and was one of the helpers for part of our workshop. And so certainly I think there'll be lots more like intro R and Python opportunities coming up on the horizon. That is something that I would love to do, is develop kind of introductory analysis modules. And so, you know, for me, I like have multiple hats on. I'm going to be teaching genomic sciences this year. And so I'm developing some material for that. And so that's something that's on my mind, is thinking about transcriptomics or single cell transcriptomics and being able to maybe offer that in the summer or the early fall as a workshop. So nothing's been decided yet. But I think that the demand is certainly there. It's the limitation right now is, you know, it's my bandwidth. So, but I think that's definitely, the demand is there. And again, if you go check out those data science guides, we have curated, you know, there are some online courses for some single cell and transcriptomics, and we've curated and said, we know these are good and we know they're correct and we would recommend them to you. So that is a place to get started. But yeah, that's what I would love to do. You could imagine in like 5 years, we have a stable of like a bunch of different workshops that we're able to kind of teach on a regular basis. You know, we're 6 months in, so it's just a timing thing. Let me, while you guys are here, if anybody's thinking about other questions, let me download my slides as a PDF and upload them real quick in the chat. It'll take just 1 second. Yeah, I think, you know, for me, I guess if you left today with only one message, my hope would be that it would be that you know that there are resources here at UAB. So whether the answer is office hours or becoming a customer or a user for the core, or whether it's like, no, actually what I'm really having trouble with is I don't know how to get started with Chiaha, and you're going to research computing, like it doesn't matter. But I think there are quite a few resources here at UAB. And so if you left with nothing else, that would be my hope that you would leave with, is knowing that there are resources and places to, if you don't know the right resource, come stop in office hours or come stop in the Slack channel, and we will help you figure out what those, where your next step should be. So the slides should be in the chat now. So let me know if you don't have, if you can't get them. Awesome, thank you. Well, that's all I got. So I'm happy to hang out if anybody has more questions, or you guys can have 10 minutes of your day back. Win-win. So
thank you again, Brittany. If you guys have any more questions, like I said, like she said, reach out in those avenues, or reach out to someone in the Informatics Club and we'll give you the links or whatever you need to do that. So thank you guys for coming. Thank you, Brittany.
You're welcome.